So my name is Andreas Jeromen, Chief Scientific Officer to Cohen Veterans Biosciences. I hope that you can hear me. You may not be able to see me, but hopefully at least you're able to hear me. So we're continuing this afternoon with the Post-Traumatic Stress Disorder Working Group of the Psychiatric Genomics Consortium, PGC. Maybe just a word about Cohen Veterans Biosciences. It's a nonprofit research organization centered around the development and diagnostics and therapeutics for trauma-related disorders. And it's one of these organization who actually has funded the global GWAS effort in PTSD, and Caroline will present us with an update. So with this, again, welcome. And we start off the presentation by Caroline Nievergelt from the University of California in San Diego about large-scale consortia efforts for PTSD. I would just say maybe from a technical perspective, people who are not speaking could probably mute themselves. I'll do the same, and then there is a chat room function. We'll have an opportunity for questions right after each presentation. So with this, Caroline, the floor is yours. Thank you.
Thank you very much, Andreas, and thank you everybody for being here. So I'm presenting on behalf of the PGC-PTSD group, and I'm addressing our current barriers to progress. And so, as you all know, post-traumatic stress disorder is unique among the mental disorders because it requires exposure to a traumatic event. And so this influences the kind of study that we have in our group compared to most of the other PGC groups that are predominantly of European ancestry. We have a much more diverse ancestry. So we have published our Freeze 2 GWAS results last year, and our analysis strategy has been to first separate the ancestry groups, then perform a GWAS. For each study, meta-analyze across studies, and then meta-analyze across ancestries finally. So highlights from our Freeze 2, where we had a total of over 30,000 PTSD cases and 170,000 controls. We performed the GWAS on 23,000 European ancestry PTSD cases, and then a smaller one on the African American ancestries. Our heritability had a large range of 5 to 20% and was higher in women. And we had 3 genome-wide significant hits, 2 in the European ancestry and 1 in the African American ancestry. And then when we did sex stratified analysis, we had 3 additional hits in the men only. So we tried to replicate our data in the Million Veteran Program Re-Experiencing Symptom GWAS, and we were not successful, but our Freeze 2 data was significantly able to predict re-experiencing symptoms in MVP. So these were our first genome-wide significant hits for the group, and as you can see, the hits are not very impressive yet, but we had some interesting candidate genes such as the PAC2 gene in the European ancestry and microRNA and the HLA-B locus in the African ancestry. So our overarching goal for the years ahead is, as everybody does, to massively increase our sample size, and then also to improve the phenotype characterization in our data. Freeze 2 had in the European ancestry over 23,000 PTSD cases, and for our next freeze, Freeze 3, we are aiming to get over 60,000 PTSD cases. So how are we going to do that? Our group is centered around working groups that deal with complementary omics data as well as phenotype data. And so led by Kirsten Kernan, we have now a new group that is working on data from electronic health records, mostly ICD-9 and ICD-10 codes to get a PTSD diagnosis. And we have at least 10 different sites that are going to contribute. So this is what we are now working on. And here is some data from the WCPG in last fall, what the different groups have presented. And so, as you can see, the schizophrenia success has extended to the other disorders, but the trajectories are very different. And so here you see PTSD in red, our Freeze 1, our Freeze 2, and then the projected Freeze 3 and 4. And we are thinking that we will have about 26 genome-wide significant risk loci in our next freeze. So what are we doing while we are working on Freeze 3? I want to give you some exciting examples. So we are optimizing the contribution of diverse ancestries to be ready when we get more data. We are harmonizing item-level PTSD and trauma phenotypes, and we are following up on our GWAS hits. So as you know, we have a lot of admixed subjects in our data. Here the example of the African Americans that are on average about 20% admixed with Europe. But as you can see, there is a wide range between the subjects, and typically we are just using PCs to adjust for population stratification. But at any given locus in the genome, a subject can have 0, 1, or 2 African tracts. And so this is information that we are trying to leverage right now. Elizabeth Atkinson in Ben Neal's lab has been developing a method called Tractor. She is to infer local ancestry and then to use this information to incorporate it into the GWAS. And from simulations that we have been doing, we can show that we are expecting to have power gains when we incorporate local ancestry information. So another example is Cathy Torres in my lab has been, over the last 2 years, tried to get item-level phenotype data from all of the studies that we have, which has been quite a challenge, but very successful now in the end. We have data from DSM-3, 4, and 5. We have PTSD assessments, lifetime and current symptoms, and we have the instruments include CAPS, PCL, and over 10 other ones. So it's quite a rich data set. And so one of the things we are doing is we are looking at quantitative PTSD symptoms right now. So the reason for that is that it's known that especially for population studies, you can get quite a substantial power gain when you look at the quantitative phenotype as opposed to case control. So Adam Mayhofer in my group has taken the data, and we have now for about 95% of the subjects PTSD symptom data. In the European ancestry, he has performed the GWAS, added the case control data where symptom data was not available, and then performed the meta-analysis and post-GWAS and MAGMA and heritability calculations. And what you see here on the top is the results that we have published in the Freeze 2 case control data with our 2 hits on chromosome 6. And we didn't have any tissue enrichment. And so compared to that, the quantitative symptom score GWAS at the bottom has 5 genome-wide significant hits, has significant tissue enrichment in the brain, and has a much more significant SNP-based heritability. So looking at the risk loci that we have now, they include some interesting genes, such as FOXP2, and we are using the MVP re-experiencing data to look at replication, and at least 3 of the 5 leading SNPs replicated nominally. So Adam has also performed a meta-analysis just recently across the quantitative symptoms in the our data and the MVP re-experiencing. And so what we found is 21 genome-wide significant hits. 8 of them were previously reported by either Joel or Murray, and I'm sure Murray is going to talk much more about this later today. We have also 30 newly identified risk loci. Some of them very interesting, such as the GABA-R1 receptor on chromosome 6. And so what we're doing now is we're looking at a little bit at the specific hits that we have found, especially in the HLA locus, which is and obviously dealing with immune dysfunction, which is a key player in PTSD. So, for example, from our analysis, we have the African-American male hit in HLA-B, and we have the this new finding that is close to the HLA-F region. And we have a secondary analysis proposal from Seema Katringli in Alicia Smith's lab, who is now imputing HLA alleles from the GWAS data with the goal to perform an association study between HLA alleles and PTSD. So she has already very interesting preliminary results, and we're looking forward to seeing that paper coming out soon. So, of course, now that we have all this phenotype data in hand, we are trying to harmonize it across the different measures, which is quite a challenge. We are trying to extract the B, C, and D clusters from the PCL4, PCL5, and matching it up with the 6 questions that were asked in the UK Biobank data. And I just want to, at the end, show you one really interesting example on how we're actually going to use this data. So, secondary analysis proposal from Mariana Gasperi at UCSD is looking at the genetic overlap between PTSD symptoms and migraine headache. And so she has 2 aims. First, she is using the twin data from the Vietnam era twin registry. She has 2,340 male twin pairs, and she's evaluating the relationship between the PTSD domains, re-experiencing avoidance and hyperarousal, and migraine headache. And then once our genomic data is becoming available, she's going to do a similar approach using genomic SEM to get at the same question using the actual genetic data. So her results from the twin data show that a bifactor model provides the best fit, with the first factor contributing to the re-experiencing, avoidance, and hyperarousal data. And the second factor is contributing to migraine, the PTSD and C clusters, but is not contributing to the re-experiencing B cluster. So as expected, the heritability and especially the genetic correlation among the PTSD clusters is very high between B, C, and D. But interestingly, the genetic correlation between migraine is in the range of around 50% for the B and D symptoms, for the C and D symptoms, excuse me, but not significant for PCLB. So with these findings, we are thinking that we can show that the relationship between migraine headache and PTSD varies across the PTSD symptom clusters, and we're looking forward to seeing these results also using GSAM. With this, I would like to thank everybody who has contributed, and I'm ready to take some questions.
Thank you, Caroline. So, Chris, I don't know, is there a different chat room? I was following this. I didn't see any questions in the chat room itself, but maybe they're captured somewhere else.
There's a Q&A button at the bottom. There's one question from Vikus Barkhuizen. Shall I just read it out to you, Caroline? So, given the interesting point made during the substance use disorder working group talk, would it be possible and interesting to restrict controls to those who have been exposed to trauma in a PTSD GWAS?
Yeah, and we have this data in hand, and we have looked into this. Since the prevalence in PTSD is not so high, it does not significantly strengthen our findings, I believe. But it's, of course, it depends on which cohort you're looking at.
And then there's a second question by Amanda Gentry asking, is there a manuscript available describing the tractor method?
The paper or the method is up on GitHub, on Elizabeth Atkinson's GitHub. The paper, we are about to submit it. Okay. So it's not available yet.
And then there's a third question. Yeah. Do you want to go on, Andrea?
You're doing a great job reading it, so I found them now. Please, go ahead.
So PTSD is highly comorbid with many other traits, like major depressive disorder. How do we differentiate the comorbid traits genetically?
Yeah, that's a very good question. We don't have much phenotype data available to do that, so we're needing to use some statistical methods for that.
So, Caroline, maybe again, to high level here, there was a recent paper in Nature Genetics, I think, by people who are actually on this call here as well, as to the low degree of specificity. I mean, what you presented today, going from overall diagnostic criteria, potentially expanding into health records now at the symptom level, the hits are not surprisingly different. I guess that's what you've showed today. I was just curious if we can expand on this into other functional domains, their data for sleep and so on. How do we now start dissecting out what PTSD really is at the biological level?
Yeah, so that is of course the goal, and yeah, how exactly we're going to do that? So the GSAM would be one of the methods we could use for that.